Hello, everyone, and welcome to today's webinar. Cell therapy needs a data-driven and certified cleanroom-compatible CO2 incubator. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Mary Kay Bates, Senior Global Cell Culture Scientist at Thermo Fisher Scientific. Mary Kay, you may now begin your presentation.
Well, thank you, Jennifer, and thanks to everyone for joining us today. Thanks again to LabRoots for welcoming me back. About a year ago, I gave a talk in this forum about the critical incubation parameters for cell therapy, how they affect cultured cells, and the effect that different incubator designs can have on culture conditions. Near the end of my talk today, I'll review a few of the points of those data that I presented a year ago. But interestingly, most of what I'm going to talk about today is about the clean room environment and how that affects the CO2 incubator, such that the incubator requires additional safeguards beyond those in an R&D lab. Also, the CO2 incubator can provide documentation that helps ease your path through regulatory audits. Now, before I go too far, I want to say that I understand that not everyone is using a CO2 incubator for cell therapy production in a clean room, but maybe this information will help you as you start to think about that transition. Over the last year, our team here has spent a lot of time thinking about this, and a lot of this information is reflected from talking directly with our clients working in clean room settings who are scaling out production. So let's begin with my agenda. I'll begin with an introduction, just reviewing the global growth of regenerative medicine. It's fun to look at the excitement in this sector. Then I'll talk about what are the needs for clean room equipment. And that involves all the sorts of not just documentation, but also cleaning and disinfection. What are the concerns for working in a clean room, and how is that different from an R&D lab? Part 3 is about what makes a CO2 incubator clean room compatible, and this is kind of the meat of the talk, and I think it's very exciting. I hope you will too. Part 4 will be just reviewing, like I said, some of the data that I presented a year ago, showing how incubator performance affects your end results because it affects how your cells grow. And then I'll finish up with some conclusions and a little summary of my main points. So, I think many of us, when we're working in the lab or the clean room, we get very focused on our work, which makes us successful. But I think it's important to take a step back now and then and look at the big picture. So cell therapy has existed for decades. The first blood transfusion occurred in 1795, and cell therapy also includes plasma, platelets, bone marrow donations, organ transplants. The first organ transplant was already way back in 1954, and I think a lot of us, of course, are thinking about vaccines a lot nowadays in this global pandemic, but vaccines, immune stimulators like interferon gamma, monoclonal antibodies, biologics like insulin, all of those can be considered cell therapy or regenerative medicine as well. The first monoclonal antibody was approved in 1986. And when we think about cell and gene therapy, I think many of us think about cancer applications in particular, but I always like to review that there are many applications in cell and gene therapy, including many applications in orphan diseases that previously really didn't have a lot of hope. So that, for me, is an inspirational point about the potential for cell and gene therapies. And all regenerative medicine applications are projected to grow in the next 5 years. cell therapies in particular, with a cumulative annual growth rate of about 27%. Interestingly, in these data, stem cell therapies are separated out, and they have kind of a comparatively low projected annual growth rate of about 9%. And if we split the growth rate into different applications, musculoskeletal disorders are actually the highest sector, and maybe that's a surprise to some people. Cancer oncology applications, as we know, are projected to have the highest growth rate at 24%, but all of these applications are projected to grow in double digits over the next 5 years. And this growth is occurring globally as regenerative medicine institutions are distributed in regions all around the world. North America does currently have the largest share of those institutions, but they are represented in every region, and that's exciting. And even in this global pandemic, which has slowed so much growth in businesses around the world, we're still seeing growth in the number of institutions around the world. So when we look at this growth, what types of institutions do we see? It may be surprising, I think, when many of us think about therapeutic institutions, we think about the large companies like Lonza, Takeda, Pfizer, but those actually represent only 8% of the total number. About 23% of these institutions are nonprofit and government academic institutions, and the remainder, the vast majority, about 64%, are startup companies and mid-sized companies, and this is where the innovation is occurring. So what's driving this growth? Well, as we see in the pandemic, diseases are increasing globally, and this includes not just respiratory viruses, but also other diseases like cancers, diabetes, heart diseases are all increasing globally. Another big part of what's driving this growth, and what we've seen play a big role in developing these vaccines for the SARS-CoV-2 coronavirus, what's driven that vaccine development so fast is collaboration. Large companies are partnering with startups, which is a great partnership. Large companies can provide the infrastructure and capability to these startup companies, where startup companies are providing the innovation that maybe large companies don't have access to so much. In addition, academics and governments are working with industry in ways that they didn't in the previous century, so that's providing a lot of innovation and new ideas. Funding is increasing. We're seeing investors take notice as they look at the growth that I just talked about and the number of institutions and the interest in applications across these sectors. We're seeing mergers and acquisitions. We're seeing licensing as large companies license the innovation from startups and so on. And finally, and maybe most importantly, government regulations are becoming easier as regulatory bodies work toward harmonization across the globe. So that provides confidence to companies so that they know they can work to the same standards regardless of what region or country they're looking for approval in. And governments are also fast-tracking approvals, and we're seeing that not just with vaccines, but also with some cell therapies. Obviously, safety and efficacy are paramount, but we can speed some of that documentation processes and some of the review processes as we've seen with the vaccines, at least in the United States and Europe. So with that, I will now turn to talking about the needs for clean room and equipment. So I always like to start with a definition of GMP, good manufacturing practices. I think many of us know that GMP stands for good manufacturing practices, but why do we have GMP? The whole goal of good manufacturing practices is to ensure that a therapeutic is safe and pure, that is effective, and that it is of high quality. And a lot of GMP is just to ensure that processes are repeatable and consistent across lots and across production lines. And I think of it as a way to remove some of the natural human variability from a system, to kind of control our natural human behavior to want to maybe skip steps. So GMP are guidelines only. They are minimum requirements that a manufacturer must meet. Every process, every company, every innovation cell therapy is different. So you know better than anybody else how to implement GMP in your institute because you know your processes better than anyone else. So these are not prescriptive rules. There can be multiple ways to comply with GMP, and GMP is typically ensured through an effective quality management system. So let's talk a little bit more about that. So the main challenges for those of us working in GMP institutions or under GMP guidelines is first, controlled documentation. We know that creating and implementing critical standard operating procedures or SOPs is a huge undertaking, and all of those are critical to your success. And as part of that, following good record keeping, good documentation procedures, okay? So because poor record keeping is often a big flag in an audit failure. It's so you have to implement. You not just have to have those SOPs and the good documentation procedures, but you'll also have to implement them. Everybody working under GMP has to know their responsibility. They have to be properly trained, and that can include repeated training, refresher training. Under quality management, it's important not just to have a quality management system, but effective implementation of those elements can make or break an audit approval. So effective implementation of a quality management system can help you detect and prevent quality issues. They help you prevent risks before they become a significant quality issue, and they help staff to evaluate a potential risk to the quality of a therapeutic process. As part of GMP, the equipment choice and acquisition is important because the equipment is the center of your production process. So equipment must be GMP compatible, and it must meet the performance criteria that were established during the development of the therapeutic in the process. So if you have great cell growth in your R&D lab, you have to be able to duplicate that great cell growth as you transition into a GMP production process. So that laboratory equipment is the center of your production, and as part of that, that equipment must be your GMP partner. So the GMP equipment, as I've already said, must meet the performance needs so that the cellular product that you produce meets the viability and quality requirements that you established in the scale-up process. The equipment must perform in a way that is reliable and repeatable. So as an example, for an incubator or a freezer, that means that you have to have fast recovery from a door opening so that the cells are in their ideal conditions as much as possible to ensure proper cell growth. If you're talking about a freezer, you want fast recovery from a door opening because even if your cells don't literally thaw or your reagents in the freezer, just warming up a little bit can affect how they freeze and can affect viability when you thaw those cells out. And that performance includes uniformity throughout the chamber so that your cells grow at the same rate or freeze at the same rate, regardless of where they exist in that incubator or that freezer. The equipment should include clear documentation. It's not just a list of specifications, but also a list of materials of composition so that you know what kinds of disinfectants you can use with that equipment. It includes performance. As I said, it needs to perform in the same way reliably and repeatably. You need to understand the maintenance that's required as you go through the process as time passes by. And everyone needs to understand the control of that equipment. You need to be able to monitor it from across the room. The equipment needs to be easy to use. Everyone using that or operating that equipment has to understand how it works, and that extends to training and refresher training. The equipment has to be easy to clean. I spend a lot of time talking about and thinking about how to make laboratory equipment easy to clean so that you spend less time maintaining your equipment and more time ensuring your process is of high quality. I mentioned clear monitoring from across the room. data logging that meshes with your building management system or your laboratory information management system. So I've mentioned a clean room a few times in this talk, so I want to review what is a clean room, because I think I have talked with a few people who say, well, of course my laboratory is clean. Of course it's a clean room. But a clean room is very different from an R&D setting. So I'm going to spend some time talking about why that is and what makes a clean room so different. So a clean room is a controlled environment. It is an isolated room with low levels of particulates. And in fact, that a clean room is defined as a room where the particle generation, retention, and introduction are all controlled. Okay, so that's important. But how is a clean room measured? How do we measure all of those particulates in a clean room? Well, this is determined by the International Organization for Standardization, or ISO. And ISO works to globally promote industrial and commercial standards. And the air quality standards that ISO establishes are based on established measured particle sizes and numbers, and they define a particle as a minute physical entity, piece of matter, with defined boundaries. So there are different levels of clean rooms, and I'm going to talk about that here. So normal indoor room air is established as ISO Class 9. And so the ISO room classes are based on a log-based 10 system. So that means that ISO Class 5 is 10 to the power of 4, or 10,000 times cleaner than normal indoor room air at ISO Class 9. And ISO Class 7 is 100 times less clean than ISO Class 5. So I hope that makes sense. And these are defined primarily looking at particles that are 0.5 micrometers and larger per cubic meter of air. Now, you may also hear about a clean room designated as grade B. That's the most commonly used in cell therapy, therapeutic production settings. A grade B clean room is defined as ISO Class 5 when at rest and ISO Class 7 when in operation. What is the difference when the clean room is at rest or in operation? The difference is people. So people are the biggest source of particulates in a clean room, as particles come off our hair, our breath, our clothing, and so people wear specialized clothing in a clean room. I guess grade A, I'll just mention grade A. Sometimes you'll hear about grade A clean rooms. A grade A clean room is ISO Class 5 at rest and in operation, and that's more difficult to achieve. So the more critical the product or process, the lower the required ISO rating. And so just as some examples, pharmaceutical production is often done in ISO Class 7, but the final fill and finish is done in ISO Class 5. So where do these particles come from in a clean room? And I mentioned that most of the particles come from us. These are, when we talk about microbial contamination, these are normal flora on our hands, our breath, our hair, and a study in 2008 demonstrated that everybody, all of us, carry about 10,000 microorganisms per square centimeter on our skin. So as we shed skin cells, we also shed those microorganisms into the environment. Microorganisms can also come from the air. Normal indoor room air at ISO Class 9 contains, on average, between 30 and 1,000 microorganisms per cubic meter of air. So this is why we work in a biological safety cabinet when we open cell culture vessels to prevent contamination entering from the air. So now I want to talk about how those particulates impact clean rooms. And cell therapy products, by their nature, represent an inherent risk due to the purification challenges. You can't purify cell therapies by filtering them because the cells themselves, that would remove the cells from the filtration process. Most particulates are much, much, much smaller than those cultured cells. So when we talk about particulate sources in the clean room, the biggest part of this pie chart, 70%, is from the people in the clean room. but 15% is coming from equipment. So how can microorganisms come from equipment when they're normal flora for us and we're the biggest source of these particulates? Well, it turns out that non-viable particulates are also a big risk in a clean room and by extension for a cell therapy. So what sorts of non-viable particles are present? These would include microscopic particles from packaging, from glass, plastic, rubber, stainless steel, hair, paint, and so on. And those are represent a high or a moderate risk to the cell therapy. A lower risk particulate, non-viable particulate, could be cell debris. It could be leftover materials from reagents in the growth media, for example. And these non-viable particulates also represent a risk to the patient if they are included in a cell therapy, which will be an injectable. And those risks include direct tissue damage from sharp little microscopic shards of glass or stainless steel as examples. They could induce an immune response or inflammation. If they get stuck in a capillary, blockage of a blood vessel for a thrombosis or a pulmonary embolism, they can also represent respiratory problems. So I want to put a number on some of these risks to the production. Why is cleanroom compatibility an important consideration for equipment? Well, here's some numbers from recent examples. So 22% of U.S. Food and Drug Administration recalls of sterile injectables between 2008 and 2012 were caused by non-viable particulates, such as those I was just talking about. And these non-viable particulates also represented the second leading cause of FDA recalls for the 10-year period between 2009 and 2019. More recently, last month in Japan, 1.6 million doses of the Moderna vaccine that were produced in Spain were put on hold due to a contamination that turned out to be bits of stainless steel from production equipment in the final film finish room that was improperly installed. So you can see that even these non-viable particles coming off of laboratory and production equipment represent a risk. They are disruptive to the treatment. They can hinder growth of your process and your company, and obviously combating those risks is very costly. So that's a lot about particles in a clean room and why the clean room is different than an R&D lab. And so I hope that this shows you why a clean room is so very much different than a standard R&D lab and why we've been thinking about how to make a clean room compatible CO2 incubator. So now I'm going to talk about what makes a clean room compatible CO2 incubator. And why would we do that? Well, I think I've just shown you a lot of reasons why. But also, cell therapy applications require new solutions that comply with these clean room standards that I was just talking about. As I said, we're shifting from an R&D to a production setting as more and more cell therapies go into clinical trials and approach approval. And the requirements are more stringent, obviously, in a clean room. We have to provide that assurance that these cell therapies produced in this equipment are safe. And that means we have a heightened focus on audits, on regulatory, on record keeping, on risk management. Also, in the clean room, because the environment is so rigorous, there's an intensive focus on cleaning procedures. I mentioned I think about this a lot in my job at this point. Documentation is required, equipment traceability, process control, and consistency across those processes, across different lots are critical. And all of this is because the end goal in this production is a treatment, a drug, or a process that will be used in a human patient whose health and safety must be assured. So at this point, I'm delighted to introduce the new Harrison Vios CR CO2 Incubator, which is part of our CTS series of laboratory equipment. This is based on our popular Harrison Vios platform that many of you may be familiar with. And this new design focuses on what is critical to clean room production, including particles. So this new design offers no disruption to the clean room environment where you're monitoring and controlling introduction, generation, and retention of particles. It also is compatible with your processes, with rigorous cleaning procedures that you use in a clean room. And all of the features have been tested and validated to ensure that it will help you through your audit process. So this incubator includes performance with superior uniformity and recovery and contamination control to help keep those cells healthy and safe. I mentioned that this is based on the existing Harrison Vios platform. This incubator focuses on cleaning. I know it's unusual to show the back of an incubator, but I think this indicates how really proud we are of this design. This design is IP54 compliant, meaning that it is resistant to ingress of dust and dirt and droplets from sprays and wipe cleaning. And it is proven compatible with dry, non-condensing VHP procedures, especially that one from Steris in particular, and I'll talk about that more in a bit. The entire exterior casing is brushed stainless steel. And the interior is completely electropolished. Also, there's documentation included with this design. Certificates and reports are included to support your validation and help you ease your way through the maintenance of this equipment. And finally, it's particle controlled, and I'll talk about that more in a minute. It is certified compatible with ISO 14644 regulations and stipulations for clean room environment. It's certified compatible with a Class 5 clean room and GMP grade A and B, even during standard operation and sterilization modes. So how do we prevent particles in the clean room environment? When I mentioned that 15% of those particles in a clean room come from the equipment in that clean room. So we've designed a new active particle controlled system where the particles in the casing are collected and passed through a HEPA filter on the rear of the incubator before that air is released to the clean room. And so you see the air coming out of that HEPA filter on the right of the right photo is indicated in a blue, showing that it's clean air released to the clean room. And then there are also 4 passive intake air vents on the back, and those are indicated also, you see the shorter blue arrows where the air is coming in to cool the electronics and replace that outgoing air. So we validated this, and how did we validate that this design is compatible with clean room use? We actually engaged a third-party independent industry specialist, TÜV in Germany, and they worked in an ISO Class 4 clean room, and their tests showed that these designs are compatible with ISO Class 5 and GMP grade A and B clean rooms. I'm not going to go into those tests too much at this point. We are going to release a white paper showing more detail on that in the coming weeks. I will say that the evaluation was conducted both during standard operation and during sterilization, and also all of the accessories for the clean room were also tested. So this just shows the certificates issued by TÜV, and you can see that they are, it says right on the certificate, certified for and compatible for Class ISO 5 clean rooms and grade A and B clean rooms. So what's all included in this incubator? One of the things we think about is, what are the questions that you have when you're maintaining the incubator and you're working to qualify it with your process? So we include an installation checklist so that you are completely informed on the best installation procedures, IQ, OQ, PQ, temperature mapping, documents, and help are available. You have the ability to confirm or correct the calibration. All of the alarms and the alerts are easy to understand, and you can test them to show during your audit process that you've worked through those alarms and everybody understands what they mean. We can help you with training on the proper use. We include a routine maintenance checklist and a spare parts list, which are critical to ensure that your equipment doesn't go down when you need it to be there for your process. And responsive customer support, of course, is a big part of ensuring that your processes are uninterrupted. Also, this incubator comes with a complete GMP documentation package to support your validation and audit needs. This is unique in the industry, and this documentation package includes technical specifications and equipment drawings. I've mentioned the preventive maintenance list and the replaceable parts, and all of the certifications to ease your regulatory audits. And also, there's test validation protocols for you to do your own testing to ensure that you understand proper operation and the quality and operation of this equipment. I mentioned that the accessories are also tested and proven compatible. We offer two sizes of this incubator, a 165-liter and a 255-liter. We've also tested them in the stacked configuration to ensure that those particles are not disruptive either. The stacking adapter, the stand, and the roller base are all compatible, and a 4 to 20 milliamp output is standard to work with your information management systems. So I mentioned disinfectant recommendations and how much I think about cleaning, and so we've done a lot of testing to ensure that commonly used disinfectants are compatible with our incubators. I do want to take this opportunity to say that I know all of you like to kind of drown your equipment in the clean room with disinfectant, but it's important to follow the manufacturer's recommendations when you use those disinfectants, and that includes the dwell time. So if I take the Spore Cleanse RTU Sterilant as an example, Spore Cleanse Steris recommends a 10-minute dwell time, and at the end of that 10 minutes, you need to remove that disinfectant, because otherwise those disinfectants build up over time. They cause pitting and corrosion of stainless steel. They can cause cloudiness of glass doors, and all of that surface roughness provides more places where microorganisms can attach. So it's important to remove those disinfectants at the end of the dwell time, and the way you do that is you follow any chemical disinfectant with 70% ethanol or 70% isopropanol to remove those residues. Finally, please do not ever use bleach to disinfect your laboratory equipment. It's well known to cause pitting and corrosion, and so these on this slide are the disinfectants that we would recommend for manual wipe disinfection of your equipment. This incubator also features a completely electropolished interior, and electropolishing offers a number of benefits. What electropolishing does is it removes the surface roughness and microscopic structure on stainless steel. And by smoothing and removing some of that microscopic structure, what that means is that it significantly reduces microscopic areas where microorganisms could attach. And it improves the corrosion resistance and eases cleaning. Now, many of you are using sterilization by vaporized hydrogen peroxide in your clean room, and we're seeing that a lot. So we wanted to make sure that our incubator was compatible with these processes. I will say that a lot of companies are offering VHP now, and not all of them are the same. Some of them are using chemicals in addition to hydrogen peroxide, such as peracetic acid, and those additional chemicals can exacerbate pitting and corrosion by the chemical. So I will say that we've looked really exclusively at the Steris procedure because the moisture is controlled. So when you have the vapor condense, because the humidity is not controlled, that increases the dwell time of that hydrogen peroxide, and that can result over time in, again, damage to the stainless steel. So we do recommend only the Steris dry, non-condensing VHP process. Another part of that is that any company that's offering this should offer you tests to confirm that the process actually worked, that you have complete elimination, complete sterilization, so complete elimination of those microorganisms that are removed by sterilization. And finally, it's important that that vapor, that gas, be neutralized and proven neutralized, because hydrogen peroxide in high concentrations is toxic to humans, and it is a known carcinogen. So that's why these procedures are performed by trained technicians wearing protective personal equipment. So not everybody is using VHP processes, so this incubator also includes an automated high-temperature sterilization, the Steri-Run sterilization cycle. This also has been fully validated by a third-party independent test lab, the Health Protection England Agency in Porton Down in the UK. We provide complete temperature maps of 48 different locations in the incubator chamber, proving that all areas reach the specified 180-degree temperature throughout the chamber. And that ensures that no colder areas remain where microorganisms could survive and grow back. Those tests performed by Health Protection England were actually done with an incubator where the time at 180 degrees was shortened from 90 minutes to 45 minutes. So this reduction of the approved indicator organizations, including not just the dry heat indicator, but also the biological indicator used for autoclave sterilization testing. And all of those were eliminated more than 6 logs, which is the requirement for testing sterilization according to the United States and European pharmacopoeias. All of those 6 logs or greater microorganisms were removed in a truncated cycle, such that the production cycle, where it holds at 80 degrees, 180 degrees for 90 minutes, results in a sterility assurance level of 12 logs, and no other manufacturer offers this level of security. But wait, there's more. We also include an in-chamber HEPA filtration. So this is a little bit different than the HEPA filtration to remove the particulates exhausted by the incubator on the back of the incubator. So this HEPA filter is in the incubator chamber, and it helps to prevent contamination that could pose a risk to your cultured cells while they're in the incubator chamber. Now, I know there's some confusion about HEPA filtration because HEPA filters are rated for their efficiency of capturing 0.3 micrometer particles. That's because, as indicated by this graph, that size at 0.3 is the hardest to catch. That's because the physical processes of diffusion, which is really efficient at catching very small particles, and impaction and interception, which are efficient at capturing larger particles, are least efficient at that medium particle size. So 0.3 micrometer particles are captured with 99.97% efficiency, but smaller and larger particles are also captured and even with greater efficiency. So this in-chamber HEPA filtration system works as a continuous dilution. So the entire chamber air volume is filtered through that HEPA filter every 60 seconds, so that within 5 minutes after a 30-second door opening, that chamber inside the incubator reaches ISO Class 5 clean room conditions. And as you can see, the air continues to get cleaner over time as the door remains closed. These HEPA filters snap in and out easily. You don't need any special tools, and there's an alarm to tell you to replace it after 12 months. All right, so finally I want to finish up talking about the incubator itself, because while I spent a lot of time talking about the clean room and why that's challenging and why we've thought a lot about that, what it really comes down to is if your cells don't grow properly, your therapy will not be successful. So I want to spend the last minutes talking about incubator performance and how that affects cell growth. So cell culture was developed with immortalized cell lines, such as the HeLa cells from Henrietta Lacks. And those cells have been growing in culture for so long, I call them Frankenstein cells, and they can survive many challenges that we continually torture them with in the laboratory. But for cell therapy, we're using cells that are much, much more sensitive. Stem cells, immune cells, and primary cells. And these cells are much more reactive to what's going on around them than these immortalized cell lines that have mutated and adjusted to these kind of artificial conditions in cell culture. So cells constantly check their surroundings. They're constantly measuring temperature, humidity, gas concentration. What are there enough nutrients in the growth medium, Am I stimulated to divide, or am I experiencing a repressor that tells me to enter senescence? Am I touching other cells, or is there room for me to multiply? And they respond to these different triggers. And so because the CO2 incubator is designed to mimic in vivo conditions, because each parameter affects cell growth, and as I just pointed out, cells respond to those changing conditions, that means that any variation in your incubation conditions can result in variation in your cell therapy in the end. So I know that there are lots of different available incubators on the market today, and it's easy to just compare a list of specifications, but performance is more than a list of specifications. So there may be different CO2 sensors, for example, that have the same specifications in terms of their accuracy, but how that sensor performs in the incubator setting, how it reacts with the temperature and the gases and air circulation and how many vessels are in the incubator at a time, all of that can be different with different incubators. So I want to spend just a few minutes looking at the different parameters provided by the incubator, how those affect cells, and why you might see differences in different incubator designs. So temperature, of course, is the place to start. And incubator temperature obviously mimics body temperature, and cells are extremely sensitive to temperature. Most cells are okay when stored for a few days at 4 degrees, so lower temperatures just kind of slow everything down, but higher temperatures are detrimental to cells, and most cells can't withstand more than a few hours of 2 degrees Celsius above their normal temperature. But for these sensitive cells that we're talking about, even a few hours of 1 degree above normal can result in cell death. So temperature is a critical component of the incubation conditions. And I'm not going to go through this too much because I talked about this a year ago, and you can find that talk online. But temperature recovery is very different depending on the design of the incubator. Different technologies produce very different conditions. And so as you see, the fastest recovery back to 37 degrees following a 30-second door opening is provided by active airflow circulation in the chamber. Carbon dioxide is the second biggest parameter provided by the incubator. And so I want to review, I think many of us don't really think about why we provide CO2. We know it's required for cell growth, and we know it's there to balance the pH of the growth medium. But this system is actually based on the physiological conditions in the bloodstream and the lung. So as this is my drawing of a blood vessel entering a lobe of a lung. And so you see that every cell in the body is putting out carbon dioxide as a waste byproduct of metabolism, such that the oxygen concentration in the lungs is pretty high. It's 5% CO2. That's different than the air that we breathe, which is currently about 0.04% CO2. So to mimic that physiological concentration, we provide 5% CO2 in the incubator. Most commonly, it really depends a little bit on what you're using for growth medium, and I'll talk about that in just a minute. Oxygen in the lungs is about 14%. I don't have time today to talk about physiological oxygen, but I do want to point out that also in the bloodstream there's a natural buffer of bicarbonate ions. And if you look at that concentration, it's equal to about 2 milligrams per milliliter of sodium bicarbonate, which is the most common buffer used in cell growth media. So if we look at that biochemistry a little bit, the buffer in the growth medium, the sodium bicarbonate, works with that CO2 gas. The buffer takes up and gives off protons to balance the pH, keeping it at a physiological neutral pH of 7.4. Now, fluctuations in the CO2 concentration can affect the morphology and growth of your cells. If the CO2 concentration is too high or too low, you'll see changes in your cells that are reflective of toxicity because the pH is either too acidic or too basic. So the control of the CO2 is critical. And the CO2 recovery to the set conditions of 5%, the most commonly used concentration, varies very differently depending on the different technologies included in that incubator. And again, I'm not going to go through this table because I've done it elsewhere, but just to point out that even different sensors that are precision can work differently depending on the incubator design and what's the airflow like in that incubator. Finally, humidity. We provide high humidity in the incubator to reduce evaporation from the growth media, because in low humidity, water is the only thing that evaporates from culture media, and as a result, the remaining salts, minerals, hormones in the growth media become concentrated such that toxic conditions can result. So just like too much salt in your diet is bad for you, maybe some minerals in your vitamins are, if you take too many multivitamins per day, that can result in toxic concentrations. That is also true for the cells which are derived from our tissues. And evaporation is 4 times faster at 80% humidity than at 93% humidity and greater. So it's important to have high humidity to prevent evaporation, but too much humidity is also bad. So humidity higher than about 95% approaches 100% humidity, and 100% humidity, of course, is rain or fog. So when the humidity is too high, that can result in condensation of that moisture into liquid droplets or even puddles in your incubator, and that condensation can provide a breeding ground for microorganisms. So proper control of humidity is critical for healthy cells. And just like temperature and CO2, humidity recovery varies greatly depending on the design of the incubator, and different technologies produce very different conditions. So we like to provide a protected integrated reservoir for the humidity in the Harrison Vios CR CO2 incubator, which provides good balance but fast humidity recovery in a dry condensation-free chamber to help provide good cell growth, but it's also very controlled. And I know that in a lot of clean rooms, an open reservoir of water like a water pan is frowned upon. So in this incubator, the water reservoir is completely covered and protected. The HEPA filter helps to pull contamination away from the water. Finally, in-chamber sensors are critical for proper recovery and accuracy of the temperature, CO2, and humidity in your cells. In-chamber sensors experience the same conditions as your cells. If you open the door, you want the sensor to react to that condition just like your cells would react. They continuously sample and react and adjust the conditions so that your cells are in their ideal conditions as much as possible, and they provide better recovery. Now, that may seem pretty expected for an incubator, but I will tell you that not every incubator includes sensors in the chamber. They might be present in a design where samples of air from the chamber are removed and passed to the sensor outside the incubator chamber. Uniformity throughout the chamber. I've talked about this a few times. Why is this important? Without good uniformity, that means that cells in different areas experience different conditions. So if you have some culture vessels in the top left or some in the bottom right of the incubator, and there isn't good uniformity, then that can result in variability in your results, and some cells may never experience the proper conditions, such that in the end your cell therapy doesn't meet your quality and characteristic analysis that is required. So I know we think about this maybe with temperature, but I also want to point out, that heat rises, but CO2 gas is heavier than air, so it sinks. So you might have different pHs in different areas of the incubator, especially as you start to use larger vessels for production. If you're controlling oxygen using nitrogen gas, you're working in hypoxic conditions for stem cell production, for example, nitrogen gas is lighter than air, so it rises. So that means that active air circulation is the only way to keep conditions uniform throughout the incubator. So I know that sometimes you might hear different things about a circulating fan and the pros and cons, but a circulating fan provides fast recovery from a door opening. It ensures uniform conditions throughout the incubator. You might hear that a circulating fan will dry out your cultures, but that really depends on the airflow design to make sure that the fan is not blowing directly on your cultures. A circulating fan also uses the opportunity to provide HEPA filtration. You can't have HEPA filtration unless you have a mechanical method for passing the air through the HEPA filter. And also a circulating fan helps to reduce condensation so that you don't have those puddles of humidity where microorganisms could survive. Well, I've reached the end. Thank you for paying attention so far. Let's just go through a couple of conclusions. As I started out in the beginning, I hope I showed you that cell therapy is projected to show double-digit growth across all applications and sectors as far as we've projected out through the year 2025. Good manufacturing practices help to ensure the safety and purity, the efficacy and quality of our cell therapies. And GMP is guidelines. It's not prescriptive rules, but those guidelines help define practices and procedures. And also documentation and record management is critical for an effective GMP process. A clean certified CO2 incubator is one that is GMP compatible. The design is proven to limit particulates that could affect your cell therapy, that could affect your clean room monitoring. Such design is also easy to maintain. It's compatible with common disinfection processes, and the accessories and the complete documentation are also compatible to help you through your regulatory audits. Finally, those design and quality components affect the performance of the incubator, and different designs affect your end results for your cell therapies. You should ask for these performance data from the manufacturer. They are available, and you should know this and how it's going to affect your process. At Thermo Scientific, we've been making CO2 incubators for many decades, and we have a history of introducing innovations to the market, as you can see on this chart. And finally, if you would like more information, please visit our website at thermofisher.com/cleanroom-CO2. Jennifer, I think I've left a few minutes for some questions, if you'd like to open those up now.
Yes, we have a few minutes for questions. So thank you, first of all, Mary Kay, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question here is, does the incubator show you the real percentage of humidity in the chamber in the display?
This particular incubator, you can control the humidity a little bit. Our goal was to provide high humidity. There's also an adjustment for lower humidity.
Okay, great. Thank you, Mary Kay. Let's see, next question. Some labs have UV lamps in the workroom to disinfect all the ambient. Is there any inconvenience with the equipment? Can UV light damage the exterior paint?
UV light does have an effect on paint. I haven't looked at that specifically, how the UV light outside the equipment would affect it. I will say that UV light inside the incubator is not very effective for disinfection because of the high humidity provided by the incubator, such that the water vapor in the air, the water droplets themselves, appear to reduce the efficacy and the reach of the UV light. That's based on a position paper from the American Biological Safety Association, where they said that the efficacy of UV light drops off precipitously in humidity above 70%. So in the clean room itself, outside the incubator, it really depends on how high the humidity is and what's the airflow to pass microorganisms across that UV light. And finally, I would say that the efficacy of UV light, like many disinfection processes, is about the exposure, what's the intensity of that UV light, and what's the exposure time. So disinfection and sterilization are time and exposure processes, right? So it's dependent not just upon the concentration, but also the exposure time. So that's something to keep in mind with those UV lights, that the UV light is at the proper intensity and that the exposure time is long enough. But yes, that can start to degrade some paints and also materials in laboratory equipment.
Thank you, Mary Kay. Our next question is, can we obtain an optional shelf of copper for effective contamination protection?
Yes, copper shelving is available for these Vios CO2, Vios CR CO2 incubators. In fact, you can get an optional 100% pure copper interior as well. I didn't mention that. Thank you for that question.
Thank you, Mary Kay. The next question. I have heard that HEPA filters are expensive to replace and require special disposal. Are these hidden costs for your cleanroom incubator?
Well, that is a common misconception. So HEPA filters inside the incubator and on the exhaust are not very expensive. I tend to equate the cost with roughly the cost of a restriction enzyme for molecular biology applications. How much that costs specifically will vary by your region and your particular vendor, but the cost is really minimal, less than a lot of accessories for incubators. And the in-chamber HEPA filter needs to be replaced only once every 12 months, and the exhaust HEPA filter we've rated for replacement every 6 months. Oh, and I should mention because filters don't release microorganisms, you can just dispose of it in your regular biological waste. That's
great. Thank you, Mary Kay. Let's see, next question. Thank you for your information on disinfectants. I see that you say 1 to 3% hydrogen peroxide as a manual disinfectant is okay. What about 6%? Would that be okay also?
I know that everybody's urge is to use higher and higher concentrations of disinfectants, but keep in mind that that can affect the quality of your stainless steel, glass, and other materials in your equipment over time. So we tend to stay with what's the lowest effective dose, and so we would probably, I guess we would absolutely recommend 3% as a maximum.
Thank you, Mary Kay. It looks like, oh, I'm sorry.
I just wanted to clarify 3% hydrogen peroxide as a manual disinfectant. Thank you, Jennifer.
Oh, sure. Thank you so much. Sorry that I interrupted. Looks like we have time for one more question. Since microbial contamination is a concern for cell therapy, would a copper-lined incubator be a good choice for a clean room?
Yes, and this kind of goes back to an earlier question where I said we do offer optional 100% pure copper interior for the Vios CR CO2 incubator. The one caveat I would say is that the copper is not compatible with VHP processes because it's a pure element. It will react with pretty much anything it touches, which is why it's so effective in controlling contamination. And as a result of that, the high concentrations of hydrogen peroxide can start to degrade the copper over time, and it can remove the protective patina that develops, which also increases the efficacy of the copper and strengthens the copper itself. So for using copper in a clean room, remember that the copper itself replaces disinfectants because it's so effective at eliminating microorganisms. But we would recommend to use 70% ethanol or 70% isopropanol just to ensure that the copper remains clean. You can use mild dish soap on a impregnated cloth in the clean room and then follow that with 70% ethanol or 70% isopropanol. But I would recommend strongly to stay away from those stronger disinfectants which are not needed for copper interiors in any case.
Thank you, Mary Kay. Do you have any final comments for our audience today?
Just thanks everyone for attending and staying this long. I know we are all busy, and especially as we work to extend our cell therapies to production setting, time is always of the essence when you're under the gun for getting things up and running. So thank you for staying this long, and I guess good luck with your developing therapeutics.
Thank you again, Mary Kay, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.